Hello, are there any questions? Okay, let me remind you, you should be working on your unknown project. If you have questions about it, how to get started, please contact me. If you don't get started right away, you might not be able to finish it in time, meaning you are running out of time. I think we're on chapter 8, is that correct? Or are we on chapter, no, we're on chapter 13. All right, you should be able to see my screen. Today is August 16th. And we're supposed to be working on chapter 13. There is a quiz this Wednesday. It'll be covering only chapter 8 and lab 9. So this quiz will cover less material than most of the other quizzes. Hopefully that'll make it a little easier for you to study for it. All of chapter 8 is covered, and then lab 9. Any questions about any of that?
I did have one question, but not about the quiz. It's kind of about a quiz. I saw on the syllabus, and I believe I remember you talking about it, but I just wanted to double check that you do take off like the lowest quiz grade, right?
Yeah, you get to drop the lowest quiz.
Okay, perfect. Thank you.
All right, let me remind you that we have no lab this week. I will be logging in from, is it 7 to 7:15? Sorry, in the summer, I never remember my hours. Yeah, I'll be logging in from 7 to 7:15 to answer any questions. If you don't have questions, you do not need to show up during those sessions. Probably, I will not even post the session unless there's a question that I really think is good, and I'll record it and post it. So there won't be any YouTube session either. But if you have questions, please log in during that time and ask them. After 7:15, if there's no further questions, I will be logging off. Any questions about that? All right, so work on the unknown project. The unknown project is due Wednesday, August 24th. And I believe your, it's not up here, but I believe your unknown notes page is closed Tuesday, August 23rd. All right, any questions about any of that? If not, I'm gonna shut that down and go to the lecture. So we were talking about viral taxonomy last time, and we covered this slide. Oop, that's not erasing. That's kind of loud. Am I kind of loud to you guys?
No.
No, I'm gonna turn that down a little because it's a little loud to me. Maybe this only controls my volume. I don't know. It might
I
not.
think it only controls your volume because when you turned it down, you sound the same.
Okay, thank you. All right, so in viral taxonomy, remember that the viral names are common English names. growing bacterial viruses in the lab, and they are called bacteriophages, is really easy. They must be grown in a cell, in a living cell. And of course, they grow in bacteria cells because these are bacterial viruses. Did I mention that when we classify viruses, that they have classifications generally of the family, and some of them, King Peter's son came over from, some of them have orders, but above that, we don't classify them because we do not know about viral evolution. Part of the reason why we don't know is that viruses are pretty small and their genomes change very rapidly. And so because of that, we don't know the evolutionary history of viruses going back, I don't know, millions of years, something like that. However, we do classify them to the cells that they infect. So we do group them by animal viruses because they infect animal cells, and plant viruses because they infect plant cells. We will be talking in this lab about bacteriophages, viruses that infect bacteria cells, and then of course, animal viruses, viruses that infect animal cells. Okay, any question about that? I'm not sure I mentioned that last time, and that's an important thing, and that is we really only classify viruses in their order, their family, the genus, the viral species, and the subspecies, and the domain that they infect. When we're growing bacteria, viruses, we generally grow them on a lawn of bacteria. And here you see the plate is largely white or off-white. And that's from the bacteria growing everywhere on the plate. And then the bacteriophages are where a lytic bacteria virus infected a cell, and then it replicates inside that cell and lyses the cell and goes out to neighboring bacteria cells, infects them, lyses them, and then that cycle continues. And what you'll see is plaques on the bacteria growth on the plate, meaning a plaque is where a virus has lysed all of the bacteria cells. And this region here has no plaques, and it's growing as a lawn of bacteria. Any question about any of that? Anyways, that's an easy way to grow a bacteria virus. Is just you plate the bacteria so it'll grow as a lawn of bacteria on the plate, and then you add a solution containing the bacteria viruses, and wherever they will attach to the cell, you'll get a plaque. Now, why this colony is smaller than this colony and that colony is that probably this colony, when I mean colony, a plaque of viruses. It's not really a colony, it's a plaque. Is that the virus initially infected the cell, lysed it, and then went out to other virus bacteria and lysed them. And so we have a big plaque. This one, the virus was plated on the agar and it didn't infect the cell initially, and it had to wait for a bacteria to grow over to the virus. And then later it infected the cell when the bacteria grew over to the virus and infected the cell. And that's why this plaque is smaller than this plaque. Any question about any of that? And then the largest plaque I can see right here, you can see that that's actually at least 3 plaques that fuse together. And what you do is you count just the number of viruses that were in solution of the suspension that you plated. Like if you plate 1 mil of a virus solution, oh, I would say this is between 200 and 300 viruses per mil. So you just count the plaques here, and that's your solution of viruses that you plated on the plate here. Any question about any of that? All right, viral multiplication. The viral genome only contains a few of the genes needed for the new virus to be synthesized. What the virus does is it hijacks the host cell and then uses that host cell to make more viruses. It takes control of the host metabolic machinery using the host ribosomes, the host tRNA, the host amino acids, the host ATP, some of the host enzymes to replicate the new viruses. The basic mechanism of viral multiplication is similar in all viruses. For bacteria viruses that we call bacteriophages, and occasionally it's shortened to phages or phages. Both pronunciations are correct. I won't use that. I'll always use the term bacteriophages. These are viruses that infect bacteria cells. They have 2 life cycles. There's the lytic life cycle, where the host cell is infected and the virus replicates, the host cell is killed, and then it's lysed, why it's called the lytic life cycle. And then the other bacteriophages can replicate using the lysogenic life cycle. In the lysogenic life cycle, the host cell remains alive. It is not lysed, it is not killed. And so the virus and the host cell, are actually living in symbiosis for as long as the lysogenic life cycle of the virus continues. Any question about any of that? So to make new bacteriophages, what do we need? What components need to be made to make a new bacteriophage? Nobody wants to take a guess? Okay. All viruses have a genome, and that has to be replicated. If it's a double-stranded DNA virus, that double-stranded DNA virus has to be replicated to get new viruses. And then the DNA has to be transcribed into messenger RNA for viral messenger RNA. And then the viral messenger RNA has to be translated to make the capsid proteins. And then the capsid proteins have to come together to make, with the viral genome, to make the new virions. So that is the minimum. Okay. Now there is an envelope on some viruses. We'll talk about how the envelope gets made later in this lesson. But this is the minimum of what must be replicated for the virus to replicate new copies of itself. You have to replicate the genome of the virus, and you have to replicate the capsid proteins. In the lytic life cycle, it has 5 stages. There's first attachment, there's then penetration, there's then biosynthesis, There's then maturation, and then that's followed by release. In attachment, the phage, that's the bacteriophage, will literally attach to the host cell and infect it. Infection happens during penetration, where the phage lysozyme, that's an enzyme that the virus has, actually opens up the cell wall. Puts a little hole in the cell wall. And then the tail sheath actually contracts, forcing the tail into the host cell. And when the virus contracts, that puts pressure on the inside of the virus, and that injects the viral genome into the cell. We'll talk a little bit more about that. And then the virus engages in biosynthesis, the production of new bacteriophage DNA and bacteriophage proteins. The virus will then mature and we'll get the assembly of the new bacteriophage particles. And then the bacteriophage will be released. What happens is one of the last things the virus does is it releases the enzyme lysozyme, and then that breaks down the cell wall. And then the host cell will have water come into the cell, and that will eventually burst the cell because there's no cell wall. Any question about any of that? All right, let's look at that in a little more detail. This is showing you an overview of attachment. The virus will literally attach to the cell. And let me blow that up a little. The virus is usually small considering the host cell that it infects. In fact, it's smaller than this. The host cell will release an enzyme, which is actually just lysozyme. And it'll be released right here, putting a hole in the cell wall right there. That's not shown here, but there it is there. And then the virus will literally contract where the sheath contracts. This is the normal virus. And you can see right here, the sheath is contracted, but puts pressure on the contents inside the cell, which is the genome. And that will inject the genome of the virus into the host cell through the tail sheath. Let me state that the attachment molecules of the virus, if the virus has a tail, the tail fibers right here and right there and right there and right there will be the place where the virus attaches to the receptors on the host cell. So these are the attachment molecules. This right here actually just releases the enzyme lysozyme, putting a hole in the cell wall. And then the sheath contracts, which injects the nucleic acid into the viral genome into the host cell. Anyways, that's attachment and penetration. We then have the genome will be replicated to make new viral genome inside the host cell. And that's the biosynthesis stage. And then the viral genome will be transcribed, making viral messenger RNA. It'll be translated into viral protein, making the capsid proteins. And we make several proteins for this virus. We have to make the proteins of the tail fibers. We have to make the proteins of the tail, the sheath, including the base plate, the pin, the proteins around the sheath, and then the proteins of the capsomere and the capsid. And then the virus will assemble, meaning the proteins will come together. The nucleic acid, the viral copies of the viral genome that have been made will be stuffed inside the head of the virus. And then the virus will self-assemble in biosynthesis. Two terms we have for lytic life cycle or burst time. That's the time from initial infection of a host cell till the time that the host bursts from lysing. So maturation, the new viruses assemble, and then the virus, the last thing it'll do is it'll code for the release. This is a viral genome encoding for lysozyme, and that will degrade the host cell wall, and that will cause the bursting of the host cell, and that will release the newly replicated viruses. Okay, so that's maturation and release. Burst time, the time from initial infection of the host cell till the time the host cell bursts. Burst size is the release of the newly replicated virions, and this will vary for the species, but it's usually between 50 newly replicated viruses to 200 replicated viruses per host cell. The burst time also will vary with the species, but it also varies with the temperature. The average is about 20 to 40 minutes, and that's probably at standard incubation temperature, which is either 35 or 37 degrees. It'll be slowed down if you use a slower temperature. All right, any question about the lytic life cycle?
I had a question about burst time. Is that from maturation to release or from attachment to release?
No, it's from attachment of the virus till release of the virus.
Okay, gotcha. Thank you.
Okay. All right, so that's the lytic life cycle. Well, the virus just replicates in the cell, lyses the cell to release the new viruses. There's also the lysogenic life cycle, and it's a bit different. Upon attachment of a bacteriophage to the virus, it will penetrate, meaning it will go through attachment and penetration, so that the viral genome of a lysogenic virus will infect the bacteria cell. But one of two things have to happen initially once the cell is infected. A lysogenic virus can enter the lytic life cycle, in which case it goes through the cycle I just discussed, which I'll show you right here. So the lysogenic virus infects the cell. It immediately decides which life cycle to begin. It can go into the lysogenic life cycle, which I'll discuss in a minute, but it can choose to go into the lytic life cycle, which case the virus genome will be replicated, the viral proteins will be transcribed and translated, the viruses will mature and assemble into new viruses, and then the virus will lyse the host cell, releasing the newly replicated viruses, which will then start the life cycle, or not the life cycle, start the virus life cycle, one of the life cycles over again. Okay, so that's the lytic life cycle. So all lysogenic viruses can enter the lytic life cycle. And they just choose, once they first infect the cell, do they enter the lytic life cycle or do they enter the lysogenic life cycle? If they enter the lysogenic life cycle, the virus lives in symbiosis with the host cell. It does not lyse the host cell for as long as the virus remains in the lysogenic life cycle. Any questions about any of that? All right, let's talk about the lysogenic life cycle in a little more detail. I think I've got a video here. Hopefully this is gonna work. I don't think it will work because I gotta get my thumb drive in here. Where is my thumb drive? I need to make that link a complete link, and then it will run for the web page, but that's not a complete link. The local link.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage.
Well, that's not good.
First, the phage particles.
Let's show
First,
on this
the
one.
phage particles stick to the surface of the bacteria.
First, the phage particles stick.
Turn down the volume on this one. One shows you can only hear it in one player, and the other one shows
Bacterial cell. They bind to specific sites called phage receptors. Next, the DNA of the phage is injected into the cell, as if from a syringe. The ends join, forming a circular molecule. Then, the injected DNA replicates inside the host bacterial cell, and phage heads and tails are synthesized. The phage DNA is then packaged into the heads, and the filled heads attached to the tails. Finally, the cell dissolves or lyses, liberating about 100 newly formed phage particles after 45 to 60 minutes.
Huh. That's not right. Well, oh, maybe this is the one I want. All right, I was running the wrong one.
Attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall. and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any question about that video? Oops, didn't mean to do that. I meant to stop the video. Oh. Instead, can you guys hear me?
Yes.
Yes.
Great, because I don't have the thumb drive. I got the thumb drive in, so. Remove that. And then I can put my headphone back in. All right. So that was the lytic life cycle and the lysogenic life cycle. Let's talk about the lysogenic life cycle in a little more detail. So the virus has infected the cell, and initially it makes the decision whether to enter the lysogenic life cycle or the lytic life cycle. This time it decided to enter the lysogenic life cycle, and the cell will remain in the lysogenic life cycle for as long as the lysogenic protein is active. There is a protein on the viral genome, which is a repressor protein, and this is the lysogenic gene. While this protein is active, it represses the lytic life cycle. So as long as that protein is active, the cell will remain in the lysogenic life cycle. Any questions about that? The phage DNA, meaning the bacteriophage DNA of a virus that enters the lysogenic life cycle, then integrates into the host cell's chromosome. You got that? So here's the viral DNA. It's a reddish purple, and the host DNA is a blue purple. The viral DNA actually integrates into the host cell's chromosome. We have a term for that. It's called a prophage, where the phage DNA is literally inside the bacterial chromosome. Now, whenever the host cell will replicate, the viral DNA will also replicate. And that is the only way that the virus replicates while the virus is in the lysogenic life cycle. So when this cell replicates its DNA, it will also replicate the viral DNA, and then each daughter cell will have a copy of the bacterial virus inside the host DNA. Any question about that? And yes, the lysogenic cell, meaning the host cell, does replicate normally. This can go on through many cell divisions where we'll have a bacteria cell that has a lysogenic virus inside it. And you can call it the symbiosis if you want. The point is this cell can go through many cell divisions and replicating where the host virus will replicate. Now, occasionally, for reasons that are not well understood, and scientists have studied this for over 70 years, and they still don't have a good handle on it, occasionally a cell that has a lysogenic virus in it will have the virus leave the lysogenic life cycle. When that happens, the prophage excises from the bacterial chromosome and will become a separate molecule of DNA. And then the virus will no longer be in the lysogenic life cycle. It'll enter the lytic life cycle, where the virus will replicate copies of its genome and make copies of the viral capsid, and then make new virions, and then lyse the cell, releasing the newly replicated virions. Any question about any of that? Now, as I stated, it isn't well understood why a lysogenic virus leaves the lysogenic life cycle and enters the lytic life cycle. It is known that the repressor protein, which maintains the lysogenic life cycle, just stops its working. It stops working, it stops being transcribed, and then the cell enters the lytic life cycle. It is known that if you damage the DNA of this cell, then that will encourage the lysogenic life cycle to enter the lytic life cycle. And it's like the virus knows that the DNA has been damaged, that this cell may die, and so it decides to not discontinue the lysogenic life cycle and enter the lytic life cycle. But all other times when the virus leaves the lysogenic life cycle and enters the lytic life cycle, we just don't know why it did. Any question about any of that? All right. There are some consequences of lysogeny. First, the lysogenic host cell will become immune to infection by the same bacteriophage, meaning the same species of bacteriophage cannot infect this cell if the cell is already infected by a lysogenic virus. Any questions about that? The other consequence is called phage conversion, that this cell here acquires new DNA, and it's the DNA of the virus that can give this cell new genes, which can give the cell new properties. This is called phage conversion, where the host cell may exhibit new properties from the genes in the bacteriophage, which are in lysogeny. One example of this is the production of the toxin to Corynebacteria diphtherium. Corynebacterium diphtherium, this bacteria only produces the Corynebacteria diphtherium toxin if the bacteria is carrying a lysogenic virus, meaning the Corynebacterium diphtherium toxin is encoded by the DNA of the lysogenic virus. So yes, depending on whether the bacteria that infects you is itself infected, will determine how sick you become. Because obviously infection by Corynebacterium diphtherium, which isn't terribly good because it's an infection, but it's not terribly bad. But it is bad if the Corynebacterium diphtherium is infected with a lysogenic virus, and then will produce the Corynebacterium diphtherium toxin. Any question about any of that? All right. Okay, enough talking about bacteriophages. What about the multiplication of animal viruses? We are trying to talk about diseases in this class, which humans get, and so therefore we need to talk about animal viruses because there are a number of animal viruses that cause disease in humans. Animal viruses follow the basic pattern of bacteriophage multiplication, but there are some differences. Like attachment is different because animal cells have no cell wall. The virus will literally attach to the cell membrane of an animal cell. Penetration is a little different too, because it's much easier entering an animal cell because they do not have a cell wall. And penetration happens by endocytosis, or fusion, and we'll talk a little bit about that later. You then have encoding of the virus, and I think that's similar to bacteriophages, that the virus has to be encoded so that the genome can be replicated and transcribed. You then have biosynthesis, and that will be similar to bacteriophages. So you have to make the genome, the nucleic acid of the virus, and you have to translate the viral protein. So obviously you're transcribing the viral genes and then making viral messenger RNA, and then that'll be translated into viral proteins. The maturation will be similar. You then have the nucleic acid of the virus get together with the capsid proteins to assemble the new viruses. And then you'll have release of the viruses. Release can be similar to a lytic bacteriophage because there are lytic animal viruses, but release can be different. And the release can follow the budding life cycle, or it can follow the lytic life cycle for animal viruses. And we'll talk a little bit more about these life cycles for animal viruses in a little bit. Any question about any of that? All right, multiplication of animal virus generally follows the same pattern of multiplication of bacteriophage viruses, but there are some differences. You should mainly know this one major difference, and that is animal viruses can replicate in the cytoplasm, but they can also replicate in the nucleus. and bacteriophages can only replicate in the cytoplasm. So that's one big difference. Any question about that? The attachment is a little different because we're not attaching to the cell wall. Animal cells don't have a cell wall. Attachment happens to the cell membrane. And it's going to be similar in that the virus will have ligands, which will be the attachment molecules, which will bind to a host cell receptor. And generally, the attachment molecules and the receptors are what's the glycoproteins? They generally are glycoproteins. You don't really need to know that, but something else that's similar is that it'll be the attachment molecule will be a molecule which is on the surface of the virus. And I suppose you could say the receptor is also going to be on the host cell will be on the surface too. Penetration will be a little different. There isn't the removal of the cell wall by the enzyme lysozyme and then contraction of the virus to inject the viral genome into the host cell. Penetration happens either by endocytosis or by fusion. Note that some earlier editions of the textbook erroneously stated that penetration happens by penocytosis. That's not correct. It's actually endocytosis, where the virus will bind to a receptor, and then the cell membrane will actually invaginate around the virus, and then this will break off, forming a vesicle, which will move the virus inside the cell, and the cell membrane will pinch off and reform. That's endocytosis, where the virus will be brought into the cell by the machinery of the host cell, endocytosis. The other way that penetration can happen is where the virus, if it has an envelope, will literally fuse to the cell membrane. Let me draw a little example of that. I don't think I need that. So the host cell is going to be big. We're just going to draw it like this. And it'll have a receptor, which I'm generally not going to draw. We're just going to draw it like that. And then the virus will attach at the receptor. So there's our virus. And it has a lipid envelope, this virus. Try and draw that here. Not too bad. And there will be a double lipid layer to the cell membrane here. Made a little mistake there, but there we go. Well, they can fuse together where the envelope of the virus will actually fuse with the cell membrane. Hopefully I can draw this. Not too good. There we go. And this one fuses here. And then that opens up the virus where the cell membrane is now like this. And then it becomes the envelope of the virus like that. It's part of the cell membrane, and then it continues this way. That opens up the virus to the cytoplasm, allowing the contents, which would be the viral genome inside the virus, can then move inside the cytoplasm. Any question about any of that? That is fusion, where the viral envelope literally fuses with the cell membrane. Let me state that if you're wondering how that happens, is that the envelope is a lipid bilayer, which the main molecule is phospholipid, which I'll try and draw this way. So there's the head of phospholipid. It's going to have two tails. Not drawing the tails very well. And then the other one will be this way. And there will be two tails this way. So the envelope mainly is made up of phospholipids, which is the main component of the cell membrane. Any question about any of that? There are other molecules in the envelope, and we'll talk a little bit about that later. But the main molecule is the phospholipids, which is the main molecule of the cell membrane. And that's how they can fuse together. It's a little more involved than that. And I don't want to save that, sorry. And we'll talk a little bit more about the, I think we'll talk about how the envelope gets formed. This, I think, is showing you endocytosis, but it would be the same if that was the envelope. It just fuses to the cell membrane, and then that opens up so that the viral genome can move into the cell. As I stated, multiplication of animal viruses follows about the same pattern as bacteriophages. One difference is release, which it doesn't have to be a difference, and that is there are lytic animal viruses which literally rupture the cell, and the virus follows the lytic life cycle. And when the virus is replicated, the new virions are assembled. The last thing the virus does is it codes for lysis of the cell. And it's not by synthesizing lysozyme. I'm not sure how a lytic animal virus lyses the cell, but it does lyse the cell. Maybe it secretes a enzyme which degrades the cell membrane. So what would that be? It would be it secretes a probably secretes a lipase, which digests the phospholipid. But I'm not sure how lytic. That's just a guess, so don't quote me on that. I'm not sure how a lytic animal virus lyses the cell. And it just occurred to me that I don't know. It's never occurred to me in many years before. But anyways, the point is an animal virus can follow the lytic life cycle, which it lyses the host cell, and then that releases the newly replicated animal viruses. Okay. Another way that an animal virus can be released from the cell is if the animal virus follows the budding life cycle. And if the animal virus follows the budding life cycle, it will have an envelope. So the envelope viruses follow the budding life cycle. What happens is the virus will replicate its viral genome and its viral capsid proteins. The two will come together, making the new virion. And usually when the virus replicates, it doesn't replicate at very great numbers. It would be replicating at only a few numbers of new viruses. And then that virus will push up against a membrane, and it will go to a membrane. Let me blow this up. That has viral proteins or glycoproteins in it. So here is a membrane, and this can be the cell membrane, it can be the nuclear membrane, or it can be the membrane of the endoplasmic reticulum. And the virus will code for proteins or glycoproteins which will be put in the membrane. And this virus will come to a region of the membrane where there are the viral proteins or glycoproteins. And then the virus will push on that membrane, budding it out, and then continue pushing until the virus rips off a piece of the membrane. And then that membrane will reanneal, meaning it'll pinch off. Come on, mouse. And this part of the membrane will fuse with that part of the membrane. This part of the membrane will fuse with this part of the membrane, so that if this is the cell membrane will reanneal. And then this part of the membrane will fuse with this part of the membrane, and that part of the membrane will fuse with that part of the membrane, making a almost like a vesicle around the virus. Of course, if this is the cell membrane, this vesicle will be outside of the cell, and we don't call it a vesicle. What we call it is a budding virus, where the virus literally breaks off a piece of the envelope or the membrane of the cell. And that membrane of the cell becomes the envelope of the virus. And that is how envelope viruses obtain their envelope. It is a piece of a membrane of the host cell. And the only part that are virus are the viral proteins and glycoproteins that are in the envelope, shown here are the circles. Okay. Any question about any of that? Now, usually when the virus follows the budding life cycle, there are not large numbers of viruses that bud out of this cell. It's only a few viruses that bud out of this cell at one time. And that way, the budding life cycle or budding virus does not kill this cell, but the cell can lose a little bit of membrane and be fine. Okay. And so the budding life cycle generally does not kill the host cell. If the host cell dies, it's because the host immune system recognizes that this cell is infected by a virus, and the host immune system mounts an immune response against this cell, and that kills the host cell. The budding virus generally, not all the time, but generally does not kill the host cell. Any question about any of that? All right, here we're seeing, I think this is a cell here, and that's a cell there, and that's probably a cell there. We're actually seeing a cell right here, as well as here, where a virus is actually budding out of it. 1, 2, 3, 4, 5, 6, 7, maybe 8 viruses are budding out of this cell. At least at this time. All right. Now, let me state that there are some naked viruses, meaning they're viruses that don't have an envelope, which are similar to the budding life cycle in that they don't bud out of the cell and grab a piece of the membrane, which then becomes a part of the envelope, but they are shed by the cell at low numbers. And how they are released from the cell is that the virus will be replicated and then the virus will be put inside a vesicle, and then the vesicle will leave the cell, releasing the newly replicated viruses. So this virus doesn't follow the lytic life cycle, and I'm going to say it's kind of following the budding life cycle. It has a different name. It's called the persistent infection life cycle. It's a naked virus that is shed from the cell at low numbers. It generally doesn't kill the cell. And it releases naked virus in an envelope. Not an envelope, in a vesicle. Any questions about any of that? You don't need to know that life cycle. You do need to know the lytic life cycle, which ruptures the cell, and the budding life cycle, which generally does not kill the host cell. At least the virus does not generally kill the host cell. And if the host cell died, it's from the immune system. All right, viruses have different genomes. There can be double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA genomes. And each of these virus types has to have a slightly different mechanism for multiplying the viral genome. Any question about any of that? For replicating the double-stranded DNA, it's pretty simpler. It just follows the normal way the cell replicates DNA, and that is the double-stranded molecule separates by a helicase into a single strand, and then that single strand is used as a template to make the other strand. And we'll talk about the other ways that the virus will replicate in a little bit. Any question about any of that? All right. You do not need to memorize this table, but we're now going to talk about DNA viruses, and we're only going to talk about DNA viruses that cause human disease. That's still not very visible. No, that's better. Well, that's very good. So there's 7 viruses of 7 families of viruses that have the genome of DNA. So we call these DNA viruses that infect human cells and cause human disease. You do not need to know all of these families, so you don't need to memorize this family. This is just showing you all the 7 families, the genera that cause the disease, and then the disease that's caused, like smallpox is caused by the orthopox virus, cowpox by the cowpox virus. We've talked a little bit about those. We'll talk a little bit more about the herpes viridae, and we've talked about them already. The herpes viruses that cause disease, such as herpes simplex virus causing herpes simplex. And there's type 1 and type 2. It's talking about how the DNA is. I'm not gonna quiz you on that. But it also tells you whether it's an envelope virus or a naked virus, and then what the capsid symmetry is, and then the size of the virus. Like I said, you do not need to memorize this table. I'm just showing you the 7 families that cause disease. I don't think we'll talk about any of the following except for maybe cowpox and smallpox, which we've already maybe talked about, and the herpes viruses. For DNA containing animal viruses, they generally replicate in the nucleus. And that's a rule of thumb. The DNA animal viruses replicate in the nucleus. Why they replicate in the nucleus is where the cell has all of the machinery for replicating DNA. And that's why DNA animal viruses replicate in the nucleus. Any question about any of that? One DNA animal virus we're going to talk about is herpes virus, herpes simplex, and that is a DNA virus. Once the viral DNA enters the host cell, one of 2 things can happen. The viral DNA can integrate into the host chromosome, similar to the lysogenic life cycle bacteriophage integrating into the host chromosome. But here we don't call it a prophage because it's not a bacteriophage, it's an animal virus, so we call it a provirus. And the other thing the DNA virus can do is that once it integrates into the host cell, the viral DNA can be expressed, making new virions, and then we go through the lytic life cycle, which is shown in the next slide. I'll talk about it in a minute. Now let's return to the provirus. If the animal virus, that's a DNA virus, integrates into the host chromosome, forming a provirus, this viral DNA will permanently become a member of the host chromosome, meaning that viral DNA will always remain in the host cell's DNA for the lifetime of the cell. And this DNA will be replicated whenever the host cell's DNA is replicated. Any question about any of that? All right. The other thing is that the viral DNA can be expressed entering the lytic life cycle. If it's a double-stranded DNA virus, the genome will be replicated, and that'll happen regardless of what the genome is composed of. The genome will be replicated, making new copies of the viral genome. The genome will be transcribed, making viral messenger RNA, which will be translated, making the capsid proteins. The capsid proteins will get together with the viral genome, making the newly replicated progeny virions. Any question about any of that? So let's look at the replication of a DNA virus in a little more detail. The virus will attach to the cell membrane, and then it'll enter the cell either by endocytosis, or if it's an envelope virus, it could enter the cell by fusion of the envelope to the cell membrane. The viral DNA will then be released into the cell. And the viral DNA, because it's a DNA virus, the viral DNA will move into the nucleus. Inside the nucleus, the viral DNA will make copies of itself. The viral DNA will also be transcribed into viral messenger RNA. The viral messenger RNA will move into the cytoplasm. Inside the cytoplasm, the viral messenger RNA will be read by the ribosomes to make viral proteins. The viral proteins will then get together with the newly replicated viral DNA. And then the virus will assemble into new virions. And then the new virions will be released from the cell. Now that can happen in multiple ways. If it's a lytic virus, it'll lyse the host cell, and that will release the newly replicated viruses from this cell. However, if it's a envelope virus, it'll be released by budding. And if it's a persistent infection virus, it'll be released in a vesicle. Any question about any of that? All right. If there's no question about that, let's move to the RNA viruses. The RNA viruses are viruses that have animal cell viruses that have a genome of RNA. There are 15 families of RNA viruses that cause infections in humans and cause disease. This table is trying to show you the different families and provide their names. We'll talk a little bit about Picorniviridae. We'll talk a little bit about Coronaviridae and Retroviridae. I don't think we talk about the others. Orthomyxoviridae, we'll briefly mention that one. And I've actually already briefly mentioned that one. That is the family of viruses that cause the flu. And if you remember, I mentioned that that virus has its genome in several segments. Not one segment, but several segments. All right, you don't need to memorize this table. It is telling you the different types of genomes they have. They're RNA, so this would be single positive. We would come down here, single negative. And there we got a double stranded RNA virus, Reroviridae. Whether it's an envelope or a naked virus, the capsid symmetry and size. And then this table, which is a little more interesting, telling you the different viruses that cause disease, like the polio virus. I think we talk a little bit about that in the future, not much, just a sentence or two. I will talk about coronavirus and retrovirus. All right. Animal containing viruses, as a rule of thumb, replicate in the cytoplasm. Well, if the animal virus has a genome composed of RNA, it almost always will be replicated in the cytoplasm. Why? Because this is where the cell has machinery for working with RNA. And this is just showing you some different RNA containing viruses, rubella, the mumps. Let me go back one page, sorry, and state there is one exception. And you do need to know that there is one exception, and that is there is one RNA virus family that does not replicate in the cytoplasm. And that is retroviruses. They are an RNA virus. They are an exception to this rule of thumb where the RNA virus replicates in the cytoplasm. Retroviruses replicate in the nucleus. And when we talk about it, you'll see why retroviruses replicate in the nucleus. And I'll just mention they have a DNA intermediate. All right, viral multiplication of RNA viruses is essentially the same for DNA viruses, but there are some differences because you have RNA instead of DNA as your genome. And that's where I was going to say viral replication occurs in the cytoplasm with the exception of retroviruses. You do have different multiplication of RNA viruses depending if you have positive strand, single strand RNA virus, or you have negative strand, single strand RNA virus. And we'll talk a little bit more about that in a little bit. For a DNA virus, this is very easy. You simply replicate each strand of DNA. For DNA, you then will use one strand of DNA if it's a double-stranded DNA molecule, and you'll transcribe that into the viral messenger RNA, and then the viral messenger RNA will be translated into a viral protein. Any question about that? If you have a genome of an RNA virus that is positive strand RNA virus, this genome itself can serve as messenger RNA and be read by the ribosome. So directly, the positive strand, single strand RNA can be read by the ribosome to make viral proteins. How the positive single strand RNA virus replicates itself is the positive single strand RNA virus makes a negative strand RNA virus, just using the positive strand as a template. And then it uses the negative strand RNA virus, if it's single stranded, as a template to make more of the positive strand, single strand RNA. And that's how positive strand RNA virus makes copies of itself, using the negative strand as a template. Now, if the genome is a negative strand RNA virus, it will copy itself by making a positive strand RNA using the negative strand as a template, and then using the positive strand RNA as a template to make more of the genomic strand, the negative strand RNA. The negative strand RNA is never translated into protein. The ribosome can't read it. And what happens is the negative strand RNA, when it makes the positive strand RNA, the positive strand RNA can act as messenger RNA and be read by the ribosome to make viral proteins. Any question about viral multiplication? Viral multiplication in animal cells? It's not shown on here, but if you had a double-stranded RNA, you would just make it single-stranded and then use one template to make your messenger RNA, or you could just use the positive strand in the double-stranded RNA to serve as messenger RNA and then translate the protein. And then to make more genome, you just use one strand as a template to make the other strand. All right, I've already talked about this, and that is if you're an animal virus and you're a negative strand, single strand RNA virus, to make more of the genome, you first make the positive strand RNA, which can be used as messenger RNA by the ribosomes to make the capsid protein. And then you use the positive strand to make more of the genomic strand. And then the genomic strand, the newly replicated genomic strand, gets together with the capsid proteins to make the new progeny virions. If you have positive strand RNA virus that's single strand, you use it as a template to make a negative strand, and then use the negative strand as a template to make more of the positive single strand RNA, which is the viral genome. Once again, only the positive RNA can serve as viral messenger RNA and be transcribed into viral protein. And then the viral protein will get together with the replicated positive genomic single strand RNA to make the progeny virions, the new viruses. Any question about any of that? Make sure I got set everything here. Yeah, I did. That's just giving you the figure. All right. In a little more detail, a positive single strand, the positive strand RNA virus will attach to a host cell. It'll be brought inside the host cell either by fusion or by endocytosis. The virus will then be disassembled. This is an RNA virus, so it'll stay generally in the cytoplasm. There is one exception with the retroviruses. The RNA is a positive strand, so it can be read directly by the ribosome to make proteins. And then the positive strand will be used to make negative strand, and then the negative strand will be used to make the positive strand, to copy the viral genome. The new viral proteins and the new viral genomes will then self-assemble. And then there will be release of the viruses by the host cell. And that could be by lysis if the virus is following the lytic life cycle. But it can also be by budding or the persistent life cycle, where the virus will break off a piece of the envelope if it's following the budding life cycle, and bud out of the cell, or it can leave the cell in a vesicle, which the naked viruses do when they multiply. All right, any question about any of this slide? Now let's move on to this slide. How does a negative antisense strand, single strand RNA virus multiply? Well, the negative single strand RNA virus attaches to the host cell, then it'll be brought into the cell by endocytosis or by fusion. The RNA will go into the cytoplasm. And it will be replicated by the negative strand making positive strand, meaning it's using the template negative strand to make positive strand, and then it's using the positive strand as a template to make more negative strand. Remember, the negative strand is the genomic strand of DNA. Any question about any of that? All right, the negative antisense single strand RNA virus will then self-assemble where the viral proteins will form around the viral genome, the newly replicated negative strand, single strand RNA. And then they will be released, the new virions will be released from the cell. That could be by the lytic life cycle, lysing the cell, or by the budding life cycle, where the newly replicated virus will bud out of the membrane of the cell. And can anyone mention the three membranes of the cell that a budding life cycle virus buds out of? Nobody's going to take a guess? All right. I'll work on that. And what else did I want to talk about on this slide? I've just forgotten what it was. Sorry. Oh, the release of the virus can happen by lysis of the host cell, or it can happen by budding, or by persistent infection, releasing the virus by a vesicle. All right, any questions? All right, the retroviruses are an exception in that they are an RNA virus, but they do not replicate in the cytoplasm. They replicate in the nucleoplasm or the nucleus. The retrovirus includes the lentivirus, including HIV-1 and HIV-2, both of which can cause AIDS. And then feline leukemia virus is also a retrovirus, and this is sometimes called kitty AIDS. It doesn't destroy the kitty's immune system, at least not until the kitty is getting ready to die. But what it does is it shuts down the immune system, and then the patient can get a number of different infections. Any question about any of that? All right, so retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. This enzyme is actually inside a virus. Let's see if I can blow that up. Up too far. Probably can't blow this up, so let me blow it up there. There we have reverse transcriptase shown by this property here. That enzyme is actually inside the virus as well as it's inside the DNA of this virus. This virus is different because reverse transcriptase is an RNA-dependent polymerase. What that means is reverse transcriptase can make DNA from an RNA template. And this is different from the rule we told you before, used to be known as a law, and that is that RNA viruses replicate in the cytoplasm, not in the nucleus. Well, reverse transcriptase is an exception, and it's because of the reverse transcriptase enzyme making DNA from RNA. This is one time where the flow of information, in fact, it's the only time I know of, where the flow of information does not move from DNA to messenger RNA to protein, or from DNA to RNA. Here is the one time when information is flowing from RNA to DNA. Retroviruses get their name from the reverse transcriptase. So R comes from reverse, E also from reverse, retro, T from transcriptase, and then R from transcriptase. So retrovirus. Attachment, penetrating, and encoding of retroviruses happens basically like other animal viruses, but biosynthesis is a different stage. In the biosynthesis of a retrovirus, which is a single strand RNA positive strand virus, the positive strand RNA virus will be made by reverse transcriptase into double-stranded DNA. And that's a two-step process. Let me diagram it for you. So here we have the viral RNA. You don't need to know this right now, but retrovirus is a positive strand, single strand RNA virus. Oh, there we go. And that's true for all retroviruses. Positive strand, single strand RNA virus. So that's what that is. Positive stranded RNA. And can I get a picture, an arrow in there? Probably not. Or you can do it, I got the wrong one. So in the presence of reverse transcriptase, see if I can make an RT. Not bad. Reverse transcriptase. We first make single strand single strand DNA. And it just uses the positive strand of RNA as a template to make the single strand DNA. And then the reverse transcriptase will take the single strand DNA and make double strand DNA. Let's see if I can copy that. Oh, there we go. This double-strand DNA will then move to the chromosome. Not a very good arrow, but good enough. Take another line here. And it'll join the host chromosome, which we're just, yeah, actually it's, I guess it's an animal cell, so we have to go like that. And how can I get a different color? We'll just go like this. And this we call, remember, a provirus. Where the genome of the virus is integrated into the genome of the, integrated into the host chromosome. So it's a part of the DNA. And this will only be replicated when the host cell replicates its DNA. And so this viral DNA will remain in the host cell and inside the host cell's DNA for as long as this host cell survives. It will never be excised. Any question about any of that? Anyways, this is explaining why retroviruses are an exception to an RNA virus, and that is retroviruses do not replicate in the nucleus. They do not replicate in the cytoplasm. They replicate in the nucleus. And the reason for that is they have this DNA intermediate. And now to get new viruses, we use the DNA to make more of the positive RNA strand, which is the genome of the virus. And then this positive RNA strand can also serve as messenger RNA to make viral capsids. And then the viral capsids will get together with the genome RNA, the replicated genome, to make the new progeny virions. So let's look at multiplication of retroviruses in a little more detail. The retrovirus actually has two strands that are both positive single-strand RNA. You don't need to know that, but you might want to remember that retroviruses is a single-strand, positive strand RNA virus. And it will infect the cell where its ligands, the attachment molecules, will bind to the receptor. And this is an envelope virus. So it'll be molecules on the envelope, most likely molecules on the spike that will attach to the receptors on the host cell. And then the the virus will be brought into the cell either by fusion of the envelope to the cell membrane or by endocytosis, where the virus will be brought into the cell in a vesicle. Once the RNA is inside the cytoplasm, reverse transcriptase that's carried inside the virus, meaning a protein that's inside the virus, will copy the positive strand, single strand RNA genome to make single strand DNA. The reverse transcriptase will then work on the single strand DNA to make it double strand DNA. The double strand DNA will then move into the nucleus. And then it will integrate into the host cell's chromosome, shown here. Let me see if I can blow that up. Sort of. And once it integrates into the host chromosome, it'll form what we call a provirus. The only time the viral DNA is replicated is when this host cell replicates its own DNA and then divides into two cells. Each will get the provirus. The provirus will be read, making more of the positive strand RNA virus, which itself can serve as messenger RNA to make new viral proteins. The positive strand, single strand RNA virus can also be replicated to make, not replicated, actually the DNA is replicated to make the positive strand RNA, which will be a new strand of the positive single strand RNA virus genome. And then the positive RNA, single strand RNA will get together with the viral proteins to make the new virus. And generally with retroviruses, they bud out of the cell. So they'll bud out of a membrane. Remember, this can be the cell membrane as shown here, but it could also be the nuclear membrane or the endoplasmic reticulum membrane. And then that will release the new viruses from the cell. And that is how the retrovirus gets its envelope. And that is, it follows the budding life cycle. It rips off a part of the membrane of its host cell. Any question about how retroviruses replicate? Remember, they are an exception. They are an RNA virus that replicates in the nucleus. And once you remember about retroviruses that they have a DNA intermediate, it should be obvious that retroviruses replicate in the nucleus, even though they are an RNA virus. All right, any question about retroviruses? If not, let's talk a little bit about Coronaviridae. These are the coronaviruses, which is the virus that is causing the pandemic, COVID-19. However, coronaviruses were well known to humans long before COVID-19 ever came around. Coronaviruses are a single positive strand RNA virus. They are enveloped. Why they are widely known, at least among virologists, is these viruses are the second most common cause of the human cold, especially in colds among children, young children. Coronaviruses cause a lot of those colds. Coronaviruses also cause SARS, MERS, and COVID-19, which are serious or cause serious diseases. SARS is severe acute respiratory syndrome, MERS, Middle East respiratory syndrome, and then COVID-19, which causes COVID-19 disease. COVID-19, if you don't know why the name is COVID-19, COVID, because it's a coronavirus, ID'd in 2019, COVID-19. That's how we got its name. This is actually a generic name. It does have a full scientific name, which is Severe Acute Respiratory Syndrome Coronavirus 2, or in short, SARS-CoV-2. And that is too close to SARS, coronavirus 1, and that's just too close in similarity to the SARS. So generally this name is not used, even among scientists. They tend to call it COVID-19 to distinguish it from SARS. Because they could call it SARS-CoV coronavirus 2. Sorry, I have trouble saying that. All right. Of these diseases, SARS, MERS, and COVID-19, MERS has the highest mortality rate, with about 36% of patients who become infected with MERS dying. But fortunately, MERS does not spread from human to human. MERS typically spreads from camels after long-term exposure to humans. And I don't know about you, but I haven't been around camels for a long, long time, okay? So apparently MERS is a virus of camels, and it can spread over from camels to people and cause MERS. SARS is the second most lethal. The mortality rate is about 10%. SARS was a virus disease that is twice broken out in China. And I think the first time was 2003, and it started spreading. There were a few cases in the United States, and a few cases in Canada, and a few cases in Japan, and a few cases in Europe. But fortunately, the healthcare facility got a hold of this disease, and they made it extinct, at least in humans. Okay. COVID-19 also first occurred in China, and fortunately it has a less mortality rate. Its mortality rate is somewhere between 0.1% and 3% of people who become infected will die. And with COVID-19, it does depend on the strain of COVID-19. You get to how the mortality rate will be. Let me see if I can remember the strain. The Delta variant of COVID-19 was the most lethal. And then the next variant after that, which I'm forgetting the name of that. I almost had it. I forget the name of that, sorry. The next variant after the Delta virus was the least lethal that we know of. It's possible that one of the new variants that have come out in the last 6 months is less lethal, but we haven't studied it for long enough, and so we don't know the lethality, or at least I don't know the lethality of the 3 newest variants. Does anybody remember the last common variant
I believe it was Omicron.
That's right, Omicron or Omicron, both pronunciations are used. That one, it was the least lethal of all the strains that I know about. Now, the last three variants, which have circulated, are all related to Omicron. So their lethality is probably low. But as I stated, they haven't been around long enough and studied long enough that they may not know. Or if somebody knows, I just don't know, because it's never been in the news yet. The problem with COVID-19 is, well, it's lethal, and then it spreads much more easily than the flu. And almost every new variety we get circulating and becoming the predominant variety on Earth spreads much more easily. So when COVID-19 first came out, it spread much more easily than the flu. And my guess now is the current variety of COVID-19 must spread something like 100 times more easier than the flu spreads. So it is very catchy, meaning it spreads very easily. And one problem with COVID-19, and one reason why we've never been able to get rid of it, is when one person becomes infected with COVID-19, they tend to infect something like 1 to 2.5 other people. Any question about any of that? COVID-19 is related to bat coronaviruses. It likely originated from bat coronaviruses, and they've sequenced the COVID-19 genome now, and they know that COVID-19 is actually a recombination of two different bat coronaviruses and a pangolin coronavirus. So COVID-19 is actually three viruses that came together sometime before 2019 and circulated among bats, and probably among at least a few pangolins before it spread to humans. What probably happened was in China, they have these markets where they sell exotic animals like pangolins, and then people buy them and then eat them or use them for medicinal purposes. And this is why a lot of the new viruses that have come up in the world have actually first appeared in China, because they have these markets where animals are in close association with people, and then the people are in large cities in China. And so once the virus crosses from the animal to the people, it then starts spreading among the people. And that is why SARS first appeared in China, and then second appeared in China. I don't remember the second time it appeared in China. And the first time was 2003. And then COVID-19 appeared in China in 2019. China has finally learned the lesson that these animal markets are a health hazard, and they're bringing viruses to humans. And so they've not shut down the markets, but they've shut down the importation of exotic animals into the markets to try and reduce the exposure of humans to new viruses. Okay, any question about that? All right. The COVID-19 virus is mostly from the two bat coronaviruses. Only about 10% of COVID-19 is from pangolin. And let me tell you, a pangolin is a spiny anteater. And the spiny anteater does have coronaviruses, which recombine to make COVID-19. And the pangolin is encoding the genome of the spikes of COVID-19. Let me show you that here, if I can blow this up. This is actually a coronavirus. And there you can see it's an envelope virus with spikes. And if you notice, this is the picture I have for my Canvas webpage. I had this long before COVID-19 ever appeared in the human population in 2019. I just thought it was a cool picture. And so I took it as my Canvas microbiology website. And now it's even more applicable because it's a picture of a coronavirus, such as COVID-19. This is what COVID-19 looks like. Any question about any of that? All right, there are different strains of the coronavirus. There was the original strain. There's alpha, beta, gamma, delta. Delta was the most lethal. Omicron, which was also called BA.1, BA.2 after that. And now we're on something like BA.4. Okay, that's the one currently spreading in the United States. This is obviously out of date. I've got here BA.2 is upticking in incidence. I guess I can change that now. Of course, it'll be out of date probably next term, but we can change that now. And I guess that needs to be changed too. Like I said, it changes all the time. And I need to change this just because I'm tired of changing this slide for whatever the current variety is. And if you haven't noticed, they're not giving the COVID-19 a name like Delta, Omicron. All of these names are letters of the Greek alphabet. I don't know where they came up with these names, but these are the type of names they're giving it now. COVID-19 is transmitted primarily via droplets from coughs and sneezes within a range of 6 feet. It can spread from indirect contact, such as when somebody sneezes on a surface and then you pick it up with your hands and then you inoculate it into your mucous membrane in your face. But surprisingly, COVID-19 tends not to spread that way. It tends to spread directly from person to person. All right, let me go back here and say, are there any questions? It's time for the class to end. Any questions? All right, if there's no questions, I will see you in the lab if you have questions after 7:15. If there aren't any more questions, I will log out. It's already 7 o'clock. Somebody was supposed to tell me at 6:50 that we were supposed to end the class. My apologies, I went a little long. All right, I'll see you in the lab.